In this lab, we're going to cover Lab 10, looking at reproduction methods. So the first thing that I would like you to do is in your lab, you want to turn to table 10-1. And you want to think about what are the advantages of asexual and sexual reproduction. So asexual means that it's reproduction without a partner. So to give you an example of that type of reproduction, that would be like bacteria, for example. Bacteria simply make a copy of themselves and they divide. There's no partner necessary. Whereas sexual reproduction involves partners. So humans, for example, do sexual reproduction. We need male, female to come together in order to reproduce for the next generation. And so what I did is on Canvas, I put in the discussion board, I broke you up into groups based on your row in the class. So the front and the back table were combined for this. And I put you in groups and I wanted you to come up with, for asexual and sexual, so for each, what are 2 advantages and what are 2 disadvantages. So again, 2 advantages for asexual, 2 advantages for sexual, 2 disadvantages for asexual, and 2 disadvantages for sexual. And so you're to go on Canvas and post your response. So you need, again, 2 of each. And once you post your response, you will be able to see other group members' response as well. And so hopefully that will get a discussion going as to what are advantages for sexual versus asexual. Why do some organisms do one type and others do a different type? Obviously, there are advantages to both types. Both types can be used and are used for organisms to reproduce. And so once you post your answers on the discussion board and you get some feedback from your group, then you're going to fill in with your answers on table 10-1, as well as question number 1 in the lab also deals with this. So use that discussion to help you guide for your answer for this question. So the first experiment that we're going to look at is looking at the spread of disease. And so this experiment is going to test the effect of multiple sexual partners on the spread of an STD through a population and how this can affect population size and growth. So again, essentially what we're going to be doing is we are going to have our class, and within our class, we're going to have one person who has the sexually transmitted disease. And so we want to see how quickly does that disease spread. So to calculate the expected number of infected individuals with an STD, assuming that only one person started with the disease, you're going to use the following formula. 2 to the n power, where n is the number of sexual partners. So again, let's say that they have 2 sexual contacts. The equation for that would be 2 to the n power, which is 2 to the 2nd power, which is 2 times 2. And we would expect that 4 people would have the disease after just 2 contacts. So if you only had 2 sexual partners, potentially 4 people could end up with the disease. Now, let's say that we have 3 contacts. 2 to the 3rd power is 2 times 2 times 2, or we would expect that 8 individuals would end up with the disease. If we have 4 contacts, 2 times 2 times 2 times 2, we would expect that 16 people would end up with the disease. And so in a minute, I'm going to show you a simulation so that you can actually see how this actually spreads through a population. And you can see that, in fact, these would be your expected numbers. And so in this activity, we want to see if we were to do this experiment and we were to start with one person with the disease, do we actually get the expected number of individuals who are infected with this disease? So let me walk you through kind of the theory behind this and then how we would actually test this if we had been in class. So here we are. So what we're looking at here is this is our classroom population. So in our classroom, we have 32 people, and we are going to simulate that only one person is going to start with this disease. And so notice that this one person who has the disease is indicated in red. So let me turn off my pen. So people in the class decide on a Friday night to go to a party. And at this party, people start pairing off and making sexual contact. And so maybe we get in the first round, these people make contact and these people make contact, and people start pairing off and having sexual encounters. Now, the person who is infected, that person is also going to make a sexual contact. So during this party, these people are going to make a sexual contact. And when that happens, we go from one person being infected to now having two people infected. And so after one contact, after the first contact, we expect that two people will be infected. The original person that had it, plus the person that they came in contact. So at this party, right, people make contact. And so you have now two people that have it. And so let's say the next night comes around and people have another party and people come in contact with other people. And so now you're going to have different sexual contact. Maybe this person has contact with this person, and these two people make contact, and these two people make contact. So now people have a second sexual encounter. So they're going to come in contact with a new partner. And so what's going to happen is that each of these people, the person who started with the disease initially and the person who contracted it, they both make a sexual contact. So that's this green line. And so after this sexual contact, now those people became infected. And notice that after two contacts, we would expect that 4 people will have the disease. And so we have our 1, 2, 3, and 4. And again, this is assuming that both people have 2 contacts. So if both people have 2 contacts, after the second contact, we would expect that 4 people would end up with the disease. And so in our simulation, we would want to see, does this actually happen? If we were to have 2 contacts, would we end up with 4 people in our population that have the disease? And so in this case, we're considering this to be a sexually transmitted disease. Let's say syphilis, for example. And so let's talk about how we would actually simulate this in our experiment. So the way that this experiment works is if we have our classroom of our 32 people, what we would do is each person would be given a test tube. And so each person has a test tube. In addition, each person would have their own bottle of fluid. And so what we would do is that bottle of fluid that you're given, you would take this transfer pipette, this plastic transfer pipette, and if you squeeze this bulb, you draw up liquid. So you would have your bottle with your fluid, you would insert your transfer pipette into the bottle, and then you would add that fluid to your test tube. Now, your test tube is going to represent your body fluids. So this is your body fluid. So all 32 people in the class will have a unique test tube. And in addition to having a unique test tube, they have a unique solution. And one person in the class is going to have the solution that simulates the disease. So in our experiment, what this actually is, and this will come into play in a minute, is that the uninfected solution is water, and the infected solution is NaOH, sodium hydroxide. So it's a base. Now, if I have a test tube and one solution has water and one solution has sodium hydroxide alone, just by looking at them, can I tell the difference between which one is which? And the answer is no. They're both clear liquids. Just like in real life, if you look at a person, I can't look at a person and say, oh, that person has HIV and that person has syphilis. You can't tell, right? Many of the diseases might have symptoms that you don't see, and therefore you can come in contact with somebody inadvertently and they could pass a disease to you because you can't tell just by looking at them that they have the disease. So our simulation is simulating that as well. You can't tell from the beginning who has the disease and who does not. The liquids look the same in your test tube. So they look the same. However, one of the solution has a base in it. And so what we would do in this experiment is all 32 people in the class will have a test tube, and 31 of them will have water in them. Only one person has the sodium hydroxide. And so what we would do is I would say, okay, now it's time to make a sexual contact. Now, the other thing that you would need to know is that each of the bottles that I would give you will have a number written on it. And so let's say, for example, that this person has bottle number 1, and this person has bottle number 2, this person has bottle 3 and 4, and let's say this person has number 5. So I simplified it. I did not include all 32, but you'll get the idea from looking at this. So when you do this, each person has a unique number. And so what I would do is I would have you guys get up, and everybody would have their test tube that has their fluid. You would still keep your transfer pipette. Your transfer pipette, think of it like your reproductive organ. So penis, vagina, whatever, that is going to represent your reproductive organ. And so I would say go, and I would set the tone with some mood music like you saw in a previous slide. And I would tell everybody, go find one other person and have a sexual contact. Now, obviously in class, you're not going to have an actual sexual contact, but the way that we would simulate this is that we would use our transfer pipette, which is our reproductive organ. Sorry about that. And what we would do is we would exchange fluids. So if these two individuals in the first round were to exchange fluids, if I was person number 1, I would use my transfer pipette, I would draw up some liquid, and I would add some liquid to my partner's tube. My partner would take some of their liquid and they would add it to my tube. And so what you're getting is you're getting this exchange of body fluids, right? The liquid inside here is representing body fluid exchange. So we would mix our fluids back and forth. I would give my partner fluids. They would give me fluids. And this is not a random hookup. You would at least have to get your partner's number or their name. And so you would record in your table for table 10-2 your first contact. So who is your first contact? So no random hookups. You have to keep track of who you made contact with. So you could write their number, you could write their name, but you have to get information for who you made contact with, okay? And so you would record who you made contact with, you exchange your body fluids, and then after you make one contact, I would say, okay, stop, don't go to the next contact until everybody has made one contact. So everybody would pair up, they would find one partner, and they would exchange their body fluids. Now, after everybody made one contact, then I would say go. I would set the mood again. And now I would instruct everybody to make a second contact. But your second contact has to be with somebody who you did not make contact with before. So let's say that these people make contact and these people make contact. So again, you would get up in the class, you would mingle throughout the classroom, you would find another student, and you would exchange body fluids. So again, you would use your transfer pipette and you would take liquid and exchange the liquids between the tubes. And so that would be your sexual contact. So you've now made 2 contacts, right? And so after 2 contacts, we would want to check to see who ends up with the disease. Now again, just by looking at the tubes, there's no way to know who has the disease. So the way that we would test to see if you have the disease after 2 contacts is that we would use phenyl red. Phenyl red is a pH indicator. And you might recall we used phenyl red back when we did our photosynthesis experiment. And so you might recall that if we blew into the phenyl red turned yellow because yellow is acidic. And so you'll recall that when we blew into the phenyl red, we were introducing carbon dioxide because we exhale carbon dioxide. CO2 was dissolving in the phenyl red, and it was forming carbonic acid, which made the pH acidic. And so phenyl red turns yellow when acidic. It also turns pink. Sorry about the pen there. When it's basic. So it turns pink when it's basic. Notice that in one of our solutions, we have our sodium hydroxide. Sodium hydroxide is a base. So after we make our contacts, if I were to come around and I were to add phenyl red to the solution, if somebody is infected, what color would their tube turn? Sodium hydroxide is a base, and so if somebody was infected, their tube would turn pink. And so I would go around the class and I would add some phenyl red to each tube. Now, the phenyl red that I would add starts out yellow. If the sodium hydroxide was in the solution, it would turn red. So when I test the person who started with the disease, their tube is going to turn that pink color. And that means that they are infected. We expect that because we know that their solution had sodium hydroxide in it, which is a base. It's going to cause the phenyl red to turn red. Now, they made contact with person number 1. And so person number 1 would have received some sodium hydroxide. And so this one would also test positive. Now, in the next round, person 1 made contact with person 3. Sodium hydroxide would be in person 3's tube. Their tube would also turn pink. And then if I go around and I test person 4 made contact with person 2 in the second round. and their tube would turn pink. Now, person number 5 over here, I put it off to the side because person number 5 had 2 contacts, but they did not have contact with anybody who had the disease. So for person number 5, all they simply did was exchange water. That is not going to make the solution become alkaline. So when I add the phenyl red to anybody who's not infected, their tube is just going to stay the yellow of the phenyl red because there's just water in there. It's not going to affect the pH, and the phenyl red is going to stay yellow. So out of our class, in this experiment with 2 contacts, we would expect that most people would have tubes that are yellow, meaning they were not infected, but we would have 4 people who would be expected to have the disease. Now, if we're doing this, what we would do at the end of this round, I would go around and I would add phenyl red to everybody's test tube to see who was infected during this experiment. If you were infected, I would have you come up and you would write your name on the board if you were infected. So if we have our list of infected, we would have person number 1, we have person number 2, person number 3, and person number 4. Now, they would write their names on the board. If we were wanting to track who started the disease, that is what's called the index patient, which I'll talk more about in a minute. So if we wanted to figure out who started with the disease, what I would do is I would say, okay, if you were somebody who tested positive, look on the board and see which numbers are up there. If both of your contacts are up on the board, that means that it's likely that you had it in the first round because both of your contacts have it, right? Person number 3 and person number 4 only have one of their contacts up there. Person number 3 made contact with person number 2. Person number 3 made contact with person number 1. Person number 4 made contact with person number 2. They would have only one of their contacts on the board. Their first contact, right, that was before they had the disease, is not going to show up on the board. And so when we do this experiment, this is how this would be done. You would have your tube. Your tube has your body fluids in it. You would take your transfer pipette. You would add your fluid into your tube. And then when I say go, you would make a contact and you would exchange body fluids with one other person. Everybody in the class would do this. Then I would say go again. Everybody would make a second contact. You would exchange the body fluids again. After the second contact, so in the first round, after the second contact is when I would add the phenyl red. And if I add phenyl red to your tube and it turns pink, that means that you are infected. You had the disease, which means you came in contact with the person whose liquid in their tube was the sodium hydroxide. So this is how this demo would work. And so we would expect to see 4 people in the class who have the disease after 2 contacts. When we do this, generally, we do have 4 people that end up with the disease. So we expect 4. Typically, we would get 4 people. So after we do this experiment with 2 contacts, the way that we would do this for the next round is that the next round is going to have 4 contacts. And so what I would have you do if this was in class is I would have you dump out this liquid. You would dump out the liquid. You would rinse your test tube and your transfer pipette. You would bring back up your original bottle. And then you would grab a new bottle. And when you grab the new bottle, you would add that to your test tube. And then we would start the demonstration over. So now everybody would have a new number. And in the next round, instead of only making 2 contacts, we would do this 4 times. And so let's look at what that would look like if we had 4 contacts. So this demonstration here is showing you if we had 2 contacts. But the idea would be the same if we're doing this for 4 contacts. The premise is the same. We would exchange our body fluids, and at the end of our 4th contact would be when I would add the phenyl red. So let's look at what that would look like. So we have our person, and our person is going to have the disease. So we set the mood again. So we set our mood, we have our party, students come to the party, and again, people are having a good time and they're starting to pair off. And so after some time, people make one sexual contact. So again, we're going to have one sexual contact. People start pairing off for their first contact. Again, we're starting with one person having the disease. After the first contact, we now have 2 people that have the disease. So after one contact, we have 2 people with the disease. The next night, we have a party, and people make a second contact. So everybody would make a second contact. So now they have their second contact. And now those people end up with the disease. So now we have 4. We have a third party. All of these 4 people are going to make another contact. And so we're going to color code that in orange. And so all of these people are going to make a third contact. So here is their third contact with the orange line. And now those people end up with the disease. So notice that we have 4 new cases. We already had 4. So after our third contact, we would have 8 infected individuals. Now we have another round, right? And so we have another night of the party, and people hook up with other people. And so now we have a 4th contact. And so notice that when this is done, we would expect that we would have 1, 2, 3, 4, 5, 6, 7, the original person, 8, 9, 10, 11, 12, 13, 14, 15, 16. So after 4 rounds, after 4 contacts, we would expect that 16 people would have the disease. So other people in the population made contact, but they didn't make contact with somebody who had the disease. The only transmission that's going to happen here is between people who come in contact with somebody else who has the disease. And so you can see that if one person in the population starts with a sexually transmitted disease and everybody in that population has 4 contacts, we would expect that 16 people in that population would end up with the disease. So just think of that for a minute. If you have 4 sexual partners, it's possible that 16 people end up with a sexually transmitted disease. And so it's just something to think about. And this is exponential. If you had 5 sexual partners, 32. 6 sexual partners, 64, and so on and so forth. And so this is just simulating how an STD would spread through a population. Now, we're going to look at what happens if our expected number does not match our actual number, because this can actually happen. And so let's look at that. So in your data for today, you can cross out table 10-2. You can cross it out because you're not actually doing the simulation. So there's no data to put here. But I will give you some example data to use for table 10-3. So round 1 is when we have 2 contacts. What was our expected number of students infected? So how many people did we expect to have the disease? And the answer is 4, right? 2 to the second power. We expected that 4 people would have the disease. Let's say that if we did this in our simulation, that 4 people have the disease. So in this situation, for 2 contacts, our expected number matches our actual. We expect 4 people have the disease, and the actual number of people is 4, right? So when I add the fennel red, 4 people have it turn up pink. In round 2, when we repeat this, and we now have 4 contacts, what is our expected number of people to be infected? Well, 2 to the 4th power, right? 2 to the 4th power. N is the number of contacts. So 2 times 2 times 2 times 2, we expect 16. So we expect 16. However, let's say when we do this, instead of getting 16, let's say our actual is only 14, right? So our actual number of people infected is less than what we expect. So now I want you to think about, why is the actual number of infected lower than expected? Why do we get less than the number of people that is expected? Well, in our simulation, one reason, and again, if you want to think about this some more, go ahead and pause it until you're ready to hear the answer, because it's always a good idea to think about this first before you listen to the answer. But when you're ready, push play. One reason that the actual might not match the expected is that 2 people are already infected, make contact. And this does not result in a new case. So let's say we have 2 people, and person number 1 caught it in the first round, and person number 2 caught it in the second round. If those people make contact in the third round, right, they both already have the disease. So if they make contact, they're not spreading the disease to 2 new people. So if that's the case, our actual number is going to be less than our expected number. And I have a slide for you in a minute to show you that, to show you actually, if you look at the population, what that would look like. But for now, 2 people that are already infected make contact. This does not result in a new case. Now, there are some other reasons that you need to think about. And some other reasons that this is the case would be, if you think about in real life, if you make sexual contact with somebody who has HIV, for example, is transmission 100%? And the answer is no. So transmission is not always 100%. And actually, females are more at risk of contracting the disease. So for a sexually transmitted disease, oftentimes females are more at risk to get the disease. But back to transmission not always being 100%. So if you make contact with somebody who has HIV, that does not necessarily mean that you are going to be infected. The reason for that is that let's say that the person who has HIV is taking antiviral medications. Those antiviral medications might suppress what we call the viral load, meaning the amount of virus that's in that patient's body. If their viral load is low enough, the risk of transmission is low, meaning they have HIV, but they might not pass it to their partner because their viral load is low enough. They don't have enough virus in the body to make it transmissible. This is also why females who are pregnant who have HIV, if they are taking their antiviral meds and their viral load is low, the risk of transmitting that from mother to fetus is actually very low. And so women who have HIV could have children who do not have HIV, because as long as they're taking those antivirals and their virus load is suppressed, that is going to help prevent transmission. Same thing with sex. If the antiviral medications are working and the virus is being suppressed, it's possible that you could have contact with somebody who has HIV and not end up with the disease. And so transmission is not always 100%. You could have contact with somebody with the sexually transmitted disease, but that doesn't necessarily mean that you are going to contract it. You're not necessarily going to become infected with it yourself. Another reason that the infected and the actual, the expected and the actual can be different could be some people are resistant to disease. So let me explain this one. So some people are resistant to the disease. This could occur naturally. In the case of HIV, viruses are obligate intracellular parasites. They must replicate within the cells. So in order for HIV to replicate, it has to get into what we call our T cells. Our T cells are a type of white blood cell, and they're used to fight off what we call adaptive immunity, meaning they target a specific pathogen. In the case of HIV, HIV has to infect T cells. And the way that that works is there is a receptor, this protein on the surface of the T cell, that the virus can bind. So the virus comes in, then it binds. So if I draw, so here's my cell. This is my T cell. And it has this receptor. Now, let's say we have HIV. HIV has surface molecules that allows it to bind. And the virus, when it binds to the receptor, can get into the T cell. And it causes the infection. And so that virus is going to hijack the T cell and turn the T cell into a virus-producing factory. And so this is how infection occurs. Now, some people can be naturally resistant to HIV. You might not know that, but some people might be naturally resistant. And the reason for that is that on the surface of their T cells, so here is the T cell of somebody who is resistant, they have inherited a mutation. They've inherited a mutation that makes their receptor look different. Their receptor is not the same as somebody who's susceptible to HIV. So if their receptor is mutated and they have this unique type of receptor, HIV can no longer bind. And if it can't bind to that person's T cell, it can't get in and it can't replicate within their T cells. And so the person is going to be resistant to HIV. And so some people are naturally resistant, right? They have this mutation in this receptor on their T cell, and it makes it so that HIV does not bind. if HIV can't bind, it can't get into the cell. And so these people are lucky, they're naturally resistant to HIV. One of the things that scientists are trying to do is to do gene modification. And that is to purposely mutate the gene that causes this receptor to be mutated. And so if they were to, let's say, vaccinate somebody with this gene mutation, right, to make it so that their genetic code changes and they have this mutated receptor, if that receptor is part of their T cells now, and now they have this mutated receptor, now they are resistant to HIV. And so that is one approach that is currently being studied, is can we genetically modify this receptor to make people resistant to HIV? So some people are naturally resistant. There was a person who was cured of HIV, and this is like a needle in a haystack. And so what happened is the person that was cured of HIV, they needed a bone marrow transplant. So when you get a bone marrow transplant, what they have to do is they have to irradiate your own blood cells, meaning they have to kill and inhibit all of your own blood cells. And so they get rid of all of the patient's own blood cells. So you actually get, sicker before you get better. So they get rid of all of your blood cells, and then they give you a bone marrow transplant. And so you have to find a bone marrow match whose genetic sequence is close enough to yours that your body is not going to reject it. And so you have to find a match. And then when you find that match, you take the bone marrow donor and you inject their bone marrow stem cells. Those are the cells that are going to populate the blood cells, meaning those are going to make T cells, B cells, etc. So the person who was cured of HIV, this is like a 1 in a million chance. This person got a bone marrow transplant. Now, you know that bone marrow transplants can be difficult to find a match, especially if you are mixed ethnicity, meaning you're a bunch of different ethnicities. It's harder to find the combination that matches you. So finding a bone marrow match is already difficult. This person got so lucky they got a bone marrow match, and to top it off, this bone marrow match was also resistant to HIV. They were also naturally resistant to HIV. So what ended up happening is when they put this bone marrow donor's stem cells into the person who had HIV, those bone marrow stem cells populated that person's immune system. So now, even if HIV was somewhere in the body, it's not going to matter because the HIV cannot infect those T cells because the T cells came from the donor who was resistant. And so this person was effectively cured of HIV because they got a bone marrow transplant from somebody who was HIV resistant. And so if you think about it, right, finding somebody who is resistant to HIV is already tricky. Finding a bone marrow match, it's tricky. But finding a bone marrow match who is also HIV resistant is like winning the lottery, right? And so this has been used and has been effective to prevent HIV. Now, in addition to some people being naturally resistant, we can also induce resistance. And what I mean by that is, depending on the disease, if you have already come in contact with that disease, right? If your immune system has seen that disease, your body is going to mount a specific immune response. Your body is going to produce what are called antibodies. These are proteins made by B cells. And when your body makes antibodies, it remembers that foreign invader. It remembers that. And so you make what are called memory B cells. So that if you were to come in contact with that again, right? If you were to come in contact with that pathogen again, you would already have resistance because your immune system has those memory B cells. and they will start producing antibodies very, very quickly, and they will make it so that you don't get the disease because your immune system will squash it before it even starts. So that is one type of way that you can acquire resistance. The other way is through immunization. And through immunization, we are purposely injecting in what we call an antigen, a marker that would be on the surface of that pathogen. Pathogen is the organism that causes disease. You inject in that antigen, and it stimulates the person to mount an immune response because your body sees what you inject in through the immunization as foreign, and your body mounts an immune response, and it makes the antibodies, and it therefore makes those memory B cells. Now, if you are immunized, that should give you protection against that disease. And so, because if you are immunized and you've seen that antigen, again, just like a natural infection, you're going to have your memory B cells. Now, you have to think about it though, and that is, do immunizations protect against everything? And the answer is no. While the chickenpox vaccine is very effective, the varicella virus, that is very effective because that virus is relatively stable. It doesn't mutate. And so once you've been immunized, you have pretty much almost lifelong protection. But if you think about the flu vaccine, right? And the flu vaccine, you have to get immunized every year. The flu mutates. And so because it mutates, it changes its antigens. And so the vaccine that we had from last year is not necessarily effective against the virus that is this year because that virus is not stable. It mutates, it changes, and now it has new surface molecules that our immune system does not recognize. And so the ability of these vaccines to protect us is quite variable depending on what we're being immunized against. Some vaccines are very effective and give you lifelong immunity. Some you might need a booster after a particular amount of time. If you think of like the tetanus shot, it's recommended that every 10 years you get a booster to boost that immunity back up. But the idea behind immunizations is that it provides resistance within the population. And one of the videos that I'm going to show you in a minute is looking at a phenomenon called herd immunity. How if most of the population is basically resistant to that disease, let's say they've been immunized, think of the measles, right? Most people in our population have been immunized against the measles. That is referred to as herd immunity. And that herd immunity is to protect the herd. And what that means is that there are people within a population who are particularly vulnerable to a disease. In the case of measles, infants have to reach a certain age before they can be immunized against measles, what's called the MMR vaccine, measles, mumps, rubella. So very young infants cannot be immunized against measles. However, if everybody around that infant has been immunized and they're all protected, then the chance of that infant contracting the disease is very low because nobody around them can be infected, which means they can't give it to the infant. And so again, that's called herd immunity. And so the idea is that if the herd is protected, then that will protect everybody. And so if you think about, the coronavirus, one of the things that in order to basically release some of the restrictions on the stay-at-home order, there are several things that have to happen in order for that to be safe. One is that you hope that your population has herd immunity. Either they've had the virus and they are now immune to it, or they are immunized against it. Now, one thing I need to say, though, is we still don't know for sure if somebody has coronavirus, if they've had the virus, are they in fact resistant? Are they in fact immune to not getting it again? Because the idea is that if we test people to see if they've ever been infected with the coronavirus, if that previous exposure gives us resistance, then those individuals who have already been exposed to the coronavirus, even if they were asymptomatic and they themselves never had it, they would still produce those antibodies. So those would be the groups of people that would be able to go back into the workforce, because assuming that infection makes you resistant to getting it again, those people would be protected. However, again, we don't know that if you've had the disease, you can't get it again, right? Again, think of the flu. You've probably had the flu multiple times. So we still don't have enough data to know for sure if that will work. But that is one idea, right? And that is that if you've already had the disease, you will have some protection against coronavirus, and therefore you could go back into the workforce without running the risk of contracting it and passing it to other people. So that's one idea. The other idea is testing, extensive testing, because the faster we track those contacts and we test to see if they are infected, the quicker we can isolate, as well as testing people to see if they've ever contracted the coronavirus. Because again, knowing that data, if we find that in fact people are resistant to the coronavirus, and we find that by tracking them and by testing people, that people have had the coronavirus, then we can assume that it is safe for those people to go out into the population. And then the last piece of the puzzle is the immunization part, right? Because if we get a vaccine to the coronavirus, then people can become artificially immune to the coronavirus, meaning that because they've injected those antigens, they've injected parts of that virus, that the person's immune system would make those B cells and would make those antibodies in the memory B cells. And therefore, if they were vaccinated, they could go into the population. And so again, I have a video in a minute to show you the effectiveness of what's called herd immunity and why herd immunity is so important for disease. Now, in terms of what we call the index case, the index case is the original infection that is introduced into a new population, right? So the original infection that was introduced into a new population. Now, when we think about the index case, as of now, current theory is that the index case, or what we call patient 0, originated in a wet market in Wuhan, China, right? And so it's thought that a bat introduced it actually into an animal called a pangolin, which then transferred it to humans. What you need to realize, though, is that while we think this is the index patient, right, based on interviews and tracking the disease, what you actually need to realize is that oftentimes we get new data that suggests that the person we called the index patient is not actually the index patient, meaning that it's possible that the disease was around longer than we knew. A really good example of this was HIV. HIV, the index patient, was always thought to have been a male homosexual flight attendant in New York. And so this was who they thought was the index patient. And as a result, there was a big stigma associated with HIV. And when HIV was originally discovered, it was thought that it was a homosexual disease, that people didn't contract it unless they're homosexual. We know that's not true. Any sex can contract or can pass and transmit HIV. It's not limited to homosexual sex. It could be heterosexual sex. Any type of sexual contact could potentially transfer HIV. And the interesting thing is while that was claimed to have been the index patient, for a very long time, the prevalent theory was it was this male flight attendant, homosexual in New York. But what we really found is that actually the index patient was this originally, HIV originated in the Caribbean in the 70s. So not in the 80s when we discovered it by this male flight attendant, but in fact, it had been around since the 70s. And so scientists estimate that HIV was transmitted to humans after a chimpanzee infected a single person sometime in the early 20th century in the sub-Saharan Africa. And then it crossed the Atlantic Ocean and spread through the Caribbean before it arrived in the US. And so possibly from Haiti, etc. And so what it's interesting to say is that, yes, we have an idea of who we think is the index patient, but in fact, that data can change. And HIV has showed us that. For a long time, people thought that the male flight attendant was the index patient. But in fact, that the disease was discovered to have actually been around in the 70s in the Caribbean. And so the index patient is based on data, but that data can change. So in this slide, I am just showing you what happens if two infected individuals make contact. So again, in our simulation, this person makes contact with one person. After the first contact, two people are infected. Then we have another round where everybody makes another contact. And after the second contact, we have 4 people who are infected. And then we do a third contact. And now we have new cases happening. So again, notice we still have 8. But what we're going to look at is in the 4th contact, what happens if 2 people who are already infected come in contact? So let's see what that looks like. So notice that this person is already infected and this person is already infected, but during the 4th round, they have contact. So if we now look at the number of cases, it is not 16. The number of cases is now 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So now only 14 people end up with the disease, even though we expect 16. And so the data that I had you guys use for that table was based on the fact that we're assuming that one time 2 people who were infected made contact and that did not result in a new case. Notice that there's no purple one coming off of this person in the red, and there's no purple one coming off this person in the orange. They did not each initiate a new case because they both already had it. And when they both already had it, that did not result in a new case. And so this is, again, just to show you that if 2 infected individuals make contact, that does not result in a new case. And our expected is not going to match our actual. Our actual number of infected individuals is less than what we would expect. If we were to do the same demonstration, again, first round, 2 people have it. Second round, they each make contact. 4 people have it. The third round, right, 4 more people have it. So we have 8. But in the 4th contact, this time we're having that 2 infected individuals make contact with one another. And so let's look at what happens there. So what we end up with is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. We end up with only 12 cases in the fourth round. And so this was just to illustrate to you that if two people are already infected and they make contact, that does not result in a new case. And that will make the number, the expected and actual, not match. The actual number will be less than what we would expect. And that's the same for any virus, right? Or any type of infection, whether it's bacterial or viral. If two people already have the disease or already had the disease and they make contact, it's not going to end up causing a new case. And so the numbers aren't always going to match. So the last part of this epidemiology simulation that I wanted to kind of talk about with you was to talk about how diseases can be transmitted, because there's been so much in the news about how diseases can be spread, meaning when we were trying to figure out with the coronavirus, how does it spread? So there are several types of transmission. We can have what's referred to as direct transmission. And in direct transmission, we have to have close contact between people. And so direct transmission examples would be things like sex, kissing. So mononucleosis is referred to as the kissing disease. It's transmitted by kissing. Touching. Droplets. So droplet direct contact. So in this case, we're talking about speech. So say it, say it, don't spray it, right? Because if you're talking and you're spitting and the organism is in your saliva, it can be transmitted. Coughing. Sneezing. Again, this is a big one for how coronavirus is shared or how it's spread. We have sharing food. Because again, if you share food and your saliva is being swapped, well, that could transmit saliva from one person to another and cause infection. Sharing food, sharing drinks. Or needles. So think of HIV, for example. HIV can be transmitted by people sharing needles. In terms of indirect transmission, in indirect transmission, an object or substance carries the agent from one person to another. And so an example of this would be fomites. And a fomite, just in case you've never heard this term, is an inanimate object that harbors and transmits the microbes. So examples of fomites would be like a doorknob, a phone, tissues, towels, bedding. So towels and bedding, for example, can spread something called pinworm, which is a worm infection. You can also transmit ringworm, which is a fungal infection, through contaminated towels or bedding. Drinking cups. Money, etc. Basically, any inanimate object that can transmit the organism from one person to another. So again, if we're talking about coronavirus is known to be transmitted through direct transmission. So again, droplets, so speech, coughing, sneezing, etc., as well as through fomites. And so touching a doorknob, that somebody who is infected has touched. And then if you touch your mucous membranes, your eyes, your nose, your mouth, etc., that can transmit it. And so this is why the importance of hand washing is so, so critical. If you go to the store, right, and you go to the grocery store and you touch things in the grocery store, you touch the cart and you touch the food items and you touch the keypad when you use your credit card. All of those types of touching, you're touching fomites. And now if you touch your face, you touch your eyes or your mouth or your nose, well, now you've transmitted potentially that microbe to yourself. And so it's really important that if you're out grocery shopping and you're touching things, while wearing gloves can be helpful if used properly, it's not necessary. As long as you practice safe, what we call aseptic technique, and that means that you wash your hands. So like if you go to the grocery store, before you get in your car, it's a good idea to use some hand sanitizer, right? So that you don't touch your car with your hands that had just been in the grocery store. So you would use some hand sanitizer and then get in the car. Now, when you come home and you unload the groceries, it's a good idea to wash your hands again. Always wash your hands. I can't stress that enough. Wash your hands, wash your hands, wash your hands. Soap and water, 20 seconds. Sing happy birthday twice. That's how long you need to be washing your hands, because anything that you touch while you're out could potentially be infectious, meaning it could have the virus on it. And if you touch it and then you touch your mucous membranes, well, you might now end up with the coronavirus as well. A vehicle. A vehicle is a natural, non-living material that can transmit infectious agents. And so in this case, it could be aerosols. And so in an aerosol, we're talking about suspensions of fine dust or moisture particles that contain the live pathogens. So a good example of a disease that's transmitted through aerosols would be TB, tuberculosis. So in the case of TB, it can pass through, so an example, we'll put air. It can survive in air currents for long periods of time. So if somebody coughs and they have TB, the organism can go into the air and it stays in the air for long periods of time. In terms of aerosols, as of now, recent data suggests that aerosols in the case of coronavirus are not as likely, meaning that the organism is not thought to persist in the air for long periods of time. So yes, if somebody were to directly cough on you or directly sneeze on you, yes. If somebody were to cough and then 10 minutes later you walk by that area, it's not likely that you're going to get the disease. And so this would be a type of aerosol. So air, so in the air, food. So in the case of foodborne illness, the organism can be found in food, and then you eat that food and now you get the disease. Soil, so infected soil. An example of this would be many of the helminths or the worms. If human fecal matter is used as fertilizer, which it is in underdeveloped countries, if human feces is used as fertilizer and that person is infected with that worm infection, they're going to transmit the eggs in their feces, and that's now going to be on the food. If you then have this organism in the soil and it ends up on the food, well, now you can end up with that worm infection as well. So soil, blood. So Ebola can be transmitted through contact with blood or body fluids. Ebola is known as hemorrhagic fever. You hemorrhage and you bleed out of different orifices in your body. And if somebody like a healthcare worker were to come in contact with that blood, It's likely to be transmitted that way. Again, and body fluids. So maybe pus from a person who's infected, etc. Parenteral is transmission via intentional or unintentional injection into deeper tissue. So again, needles. So let's say a doctor is taking a blood sample from a patient, right? And they happen to accidentally puncture their glove and inject themselves. That would be an example of a parenteral type of transmission. It's from a needle. It's inadvertently. They didn't mean to do it, but it can still transmit the disease. Knives. Any sharp, like a nail, right? So tetanus, for example, can be found on rusty knives or nails. And if you happen to get punctured with that and the organism gets in, you could end up with tetanus, which causes lockjaw. We can have branches. So there's a type of fungus that can be found on the thorns of roses. And if you happen to prick yourself with that thorn of the rose and that fungus gets into the wound, it causes something called rose gardener's disease, and it causes an infection where it penetrated the skin. We could have broken glass, etc. And so different diseases have different modes of transmission. And in some cases, diseases can be transmitted through multiple types of ways, multiple ways that they can be transmitted. Now, we can also have what are called vectors. And a vector is a living organism that's infected with a microbe and transmits the disease. And so this living organism is going to transmit the disease. So examples of this, mosquitoes. So malaria is transmitted by mosquitoes. Biting flies. So in Africa, they have a type of fly called a tsetse fly that causes African sleeping sickness. So it injects the organism when it takes a blood meal. Ticks. So if you know about Lyme disease is transmitted through the bite of a tick. Fleas. So basically any type of biting arthropods can cause this. Bats, dogs, etc. Those bats and dogs can transmit rabies, right? The organism is infected with that virus, and if they bite you, they can transmit it to you. We call that a biological vector. Some animals or some organisms are not biological vectors. They are referred to as mechanical vectors. And that is that the living organism is not infected, but carries and transmits the pathogens. So the animal itself is not infected, but the organism is somewhere on their body, and it's just physically transmitted by touch from one place to another. So an example of this would be like a housefly. So let's say a housefly, right? It lands on feces and it picks up bacteria from feces, and then that fly lands on your food and it transmits that bacteria from the feces onto your food. That's a mechanical vector. The fly itself was never infected, but on its feet where it lands, that part had touched bacteria and then landed on your food. And so houseflies, cockroaches, cat or bird scratches. Sorry, my pen's acting a little funny. So cat claws can have an organism called Bartonella on it. And if they scratch you, it can cause what's called cat scratch fever. The cat itself is not infected, but the organism is on its claws. And if they scratch you, it can get into your body that way. So that would be a mechanical vector. And so what you can see is there are a variety of different ways that diseases can be transmitted. There's not just one way. Different diseases have different modes of transmission. So what we're going to look at for this is I have this video, and it's going to talk about herd immunity and talk about how diseases can be transmitted. And so I want you to watch this video and just kind of see kind of how this, how herd immunity can offer protection. And so I'm going to play this video for you now.
Welcome to this McMastery Coronavirus Update. I'm Franz Wiesbauer. I'm an internist trained in epidemiology and public health at Johns Hopkins and the founder of McMastery, where we teach important clinical skills to doctors and other healthcare providers around the world. Today we're going to talk about herd immunity and how to stop an epidemic. Let's get started. So what is herd immunity? Well, it's a basic principle that's used in combating epidemics. Herd immunity occurs when a significant proportion of the population or the herd have been vaccinated or are immune by some other mechanism, resulting in protection for susceptible individuals. Let's see how this works in action. In a previous video, we talked about R0, the basic reproductive ratio. Let's look at this example where we have 1 diseased or infected individual and 18 susceptible individuals. As you might remember, R0 is the average number of individuals an infected person gives the disease to. R0 is fairly constant for a given disease. R0 for the novel coronavirus SARS-CoV-2 has been estimated to range somewhere between 2 and 3. So let's say this COVID-19 patient gives the disease to these 3 individuals. And let's say these 3 individuals give the disease to 3 other individuals in turn. Then this is what the situation will look like after some time. Now let's assume that we vaccinated some of these susceptible populations such that they became immune to the virus. Now the virus can't infect the individuals it infected in the previous scenario. Now our index case only infects 1 other person. And due to the immunity or herd immunity of the group, this newly infected case can also only infect 1 other person. What we've done here is to reduce the basic reproductive ratio of 3 to an effective reproductive ratio of 1. As we've seen in a previous video, when R is equal to 1, the disease remains stable and won't grow. The herd immunity threshold is the proportion of a population that need to be immune in order for an infectious disease to become stable in that community. Or in other words, in order for R to become equal to or lower than 1. If this is reached, for example through immunization, then each case leads to a single new case and the infection will become stable within that population. So how do we know what proportion of the population needs to be immunized in order to reach herd immunity? Let's look at a disease with an R0 of 8, where 1 infected individual infects 8 others on average. What would need to happen for them to only be able to infect 1 other person? Well, we would need to immunize these 7 over here. So 7 divided by 8, or 7/8, is the herd immunity. It's calculated as R0 minus 1 divided by R0. As we've learned previously, R0 for SARS-CoV-2 is between 2 and 3. So how many people would we have to vaccinate if there was a vaccine in order for the epidemic to stop? Well, that's 2 minus 1 divided by 2, which is 1/2 or 50% if R0 was assumed to be equal to 2, and 3 minus 1 divided by 3, or 2/3 if R0 was assumed to be equal to 3. So according to this calculation, we'd have to vaccinate between 50 and 66% of the population. Now, I recently heard Mark Lipsitch, an epidemiologist from Harvard, mention in a podcast interview that the herd immunity of COVID-19, according to his data, was around 40%. I'm sure he has more complex tools to factor in other variables that could influence herd immunity. But if you want to go by the books, the calculation of herd immunity according to the formula we provided is valuable and valid. By the way, I really recommend you follow Mark on Twitter. He provides great insights about the epidemic and seems to be a super smart guy. That's it for now. If you want to improve your understanding of key concepts in medicine and improve your clinical skills, make sure to register for a free MedMastery trial account, which will give you access to free videos, downloads, and updates. We'll help you make the right decisions for yourself and your patients. If you like this video, make sure to subscribe to our YouTube channel so you'll get notified when we publish new videos. See you soon.
So what that shows you is that, in fact, if we think about why herd immunity is so important, a recent example of this would be to think about the outbreak of measles in 2017. You might recall hearing about the measles outbreak at Disneyland. Now, measles was once thought to be eradicated from our country. However, because of this big boom in not vaccinating children, for example, because there was a study by Andrew Wakefield who claimed that vaccines cause autism. He later retracted that claim. But as a result of that claim, there was this social movement to not vaccinate. And as a result, that resulted in a measles outbreak. Because if we had had herd immunity, if most everybody had been immunized, the outbreak could not have occurred because most of the people who got it had not been immunized. Now, there were a few instances of people who had been vaccinated who ended up getting it, but as a general rule, most people who got the measles during that outbreak were people who were not vaccinated. If we had had herd immunity and most people in the population had been immunized, that would not have occurred. And so that has happened in several regions where a lot of people are anti-vaccine and they live together, and they've also had outbreaks of the measles as well. And so this is just showing you the importance of herd immunity. What this also illustrates is that this is partly why we are currently doing social distancing, right? Because the idea is that we want to wait for people to start making their everyday contacts until either enough of the population has had it and we can demonstrate that that gives immunity, right? So again, that's where testing comes into play. If we can show that people tested positive for having coronavirus at some point and they make those antibodies and they are protected, then they can go back into the general population. If enough people have either contracted it or have been immunized for it, so if and when we develop a vaccine for the coronavirus, if we then have that herd immunity, at that point, it'll be safe to resume more normal contact. Until that point, until we reach that stabilization, we're going to still continue to have more and more and more cases. And so again, this is just showing you the importance of herd immunity. So I have one more video that will show you about the coronavirus that just gives you a little bit more about how it's transmitted. And so I like this to demonstrate and to give you some more insight about the coronavirus itself.
In December 2019, the Chinese authorities notified In December 2019, the Chinese authorities notified the world that a virus was spreading through their communities. In the following months, it spread to other countries, with cases doubling within days. This virus is the severe acute respiratory syndrome-related coronavirus 2 that causes the disease called COVID-19 and that everyone simply calls coronavirus. What actually happens when it infects a human, and what should we all do? A virus is really just a hull around genetic material and a few proteins, arguably not even a living thing. It can only make more of itself by entering a living cell. Corona may spread via surfaces, but it's still uncertain how long it can survive on them. Its main way of spreading seems to be droplet infection, when people cough, or if you touch someone who's ill and then your face, say rubbing your eyes or nose. The virus starts its journey here and then hitches a ride as a stowaway deeper into the body. Its destinations are the intestines, the spleen or the lungs, where it can have the most dramatic effect. Even just a few coronaviruses can cause quite a dramatic situation. The lungs are lined with billions of epithelial cells. These are the border cells of your body, lining your organs and mucosa, waiting to be infected. Corona connects to a specific receptor on its victim's membranes to inject its genetic material. The cell, ignorant of what's happening, executes the new instructions, which are pretty simple: copy and reassemble. It fills up with more and more copies of the original virus until it reaches a critical point and receives one final order: self-destruct. The cell sort of melts away, releasing new corona particles, ready to attack more cells. The number of infected cells grows exponentially. After about 10 days, millions of body cells are infected and billions of viruses swarm the lungs. The virus has not caused too much damage yet, but corona is now going to release a real beast on you, your own immune system. The immune system, while there to protect you, can actually be pretty dangerous to yourself and needs tight regulation. And as immune cells pour into the lungs to fight the virus, corona infects some of them and creates confusion. Cells have neither ears nor eyes. They communicate mostly via tiny information proteins called cytokines. Nearly every important immune reaction is controlled by them. Corona causes infected immune cells to overreact and yell bloody murder. In a sense, it puts the immune system into a fighting frenzy and sends way more soldiers than it should, wasting its resources and causing damage. Two kinds of cells in particular wreak havoc. First, neutrophils, which are great at killing stuff, including ourselves. As they arrive in their thousands, they start pumping out enzymes that destroy as many friends as enemies. The other important type of cells that go into a frenzy are killer T cells, which usually order infected cells to commit controlled suicide. Confused as they are, they start ordering healthy cells to kill themselves too. The more and more immune cells arrive, the more damage they do, and the more healthy lung tissue they kill. This might get so bad that it can cause permanent, irreversible damage that leads to lifelong disabilities. In most cases, the immune system slowly regains control. It kills the infected cells, intercepts the viruses trying to infect new ones, and cleans up the battlefield. Recovery begins. The majority of people infected by corona will get through it with relatively mild symptoms. But many cases become severe or even critical. We don't know the percentage because not all cases have been identified, but it's safe to say that there is a lot more than with the flu. In more severe cases, millions of epithelial cells have died, and with them, the lung's protective lining is gone. That means that the alveoli, tiny air sacs via which breathing occurs, can be infected by bacteria that aren't usually a big problem. Patients get pneumonia, respiration becomes hard or even fails, and patients need ventilators to survive. The immune system has fought at full capacity for weeks and made millions of antiviral weapons. And as thousands of bacteria rapidly multiply, it is overwhelmed. They enter the blood and overrun the body. If this happens, death is very likely. The coronavirus is often compared to the flu, but actually, it's much more dangerous. While the exact death rate is hard to pin down during an ongoing pandemic, we know for sure that it's much more contagious and spreads faster than the flu. There are two futures for a pandemic like corona: fast and slow. Which future we will see depends on how we all react to it in the early days of the outbreak. A fast pandemic will be horrible and cost many lives. A slow pandemic will not be remembered by the history books. The worst-case scenario for a fast pandemic begins with a very rapid rate of infection because there are no countermeasures in place to slow it down. Why is this so bad? In a fast pandemic, many people get sick at the same time. If the numbers get too large, healthcare systems become unable to handle it. There aren't enough resources like medical staff or equipment like ventilators left to help everybody. people will die untreated. And as more healthcare workers get sick themselves, the capacity of healthcare systems falls even further. If this becomes the case, then horrible decisions will have to be made about who gets to live and who doesn't. The number of deaths rises significantly in such a scenario. To avoid this, the world, that means all of us, needs to do what it can to turn this into a slow pandemic. A pandemic is slowed down by the right responses, especially in the early phase, so that everyone who gets sick can get treatment and there's no crunch point with overwhelmed hospitals. Since we don't have a vaccine for corona, we have to socially engineer our behavior to act like a social vaccine. This simply means two things: not getting infected and not infecting others. Although it sounds trivial, the very best thing you can do is to wash your hands. Soap is actually a powerful tool. The coronavirus is encased in what is basically a layer of fat. Soap breaks that fat apart and leaves it unable to infect you. It also makes your hands slippery, and with the mechanical motions of washing, viruses are ripped away. To do it properly, wash your hands as if you've just cut up some jalapenos and want to put in your contacts next. The next thing is social distancing, which is not a nice experience, but a nice thing to do. This means no hugging, no handshakes. If you can stay at home to protect those who need to be out for society to function, from doctors to cashiers or police officers. You depend on all of them. They all depend on you to not get sick. On a larger level, there are quarantines, which can mean different things, from travel restrictions or actual orders to stay at home. Quarantines are not great to experience and certainly not popular, but they buy us, and especially the researchers working on medication and vaccination, crucial time. So if you are put under quarantine, you should understand why and respect it. None of this is fun, but looking at the big picture, it is a really small price to pay. The question of how pandemics end depends on how they start. If they start fast with a steep slope, they end badly. If they start slow with a not so steep slope, they end okayish. And in this day and age, it really is in all of our hands, literally and figuratively. A huge thanks to the experts who helped us on short notice with this video, especially Our World in Data, the online publication for research and data on the world's largest problems and how to make progress solving them. Check out their site. It also includes a constantly updated page on the corona pandemic.
And so again, that video just illustrated what you've heard so much about flattening the curve, and that is to try and make it so that the infection is a slow infection, so that we don't overburden the healthcare system. So the next part of the lab is looking at something referred to as alternations of generations. And so what alternations of generations refers to is it refers to this alternation of stages between the diploid part of the life cycle, and the diploid part is referred to, let me write this on here. The diploid part of the life cycle is called the sporophyte generation. And the haploid part, the part that is haploid, meaning they have half of their chromosome number, is referred to as the gametophyte. Sorry, the pen's being a little funky. The gametophyte generation. And so again, notice this is haploid, this is diploid. So the diploid is called the sporophyte generation, and the haploid phase is called the gametophyte. Think of gametes, right? Gametes are haploid, back to when we talked about meiosis. So this would be the haploid part of the life cycle. So if we look at, we're going to look at alternations of generations in plants specifically. And so if we look at plants, remember back to our diversity lab, that the most primitive plants on land will be the mosses, the bryophytes. And so in mosses, those are our primitive plants. Remember that they lack a vascular system and therefore they grow low to the ground. These primitive plants, these mosses, they are gametophyte dominant. And of the plants, they are the only ones that are gametophyte dominant. The other plants, so the ferns, the conifers, the flowering plants, those are all sporophyte dominant. However, the mosses, these primitive plants, are gametophyte dominant, which means that the bulk of the plant is actually the gametophyte generation. So notice that in this case, what we call the leaves in a moss is haploid. Those cells are actually haploid. This top structure, which is called the sporangium, is going to be the diploid part. But the bulk of the plant in the moss is part of the gametophyte generation. That's in contrast to if we look at ferns. Notice that ferns say diploid dominant. The bulk of the plant, including the leaves, is going to be sporophyte generation. The gametophyte generation is a much smaller proportion. It has this prothallus, which is this little, it almost looks kind of like a patty that a frog would get on. It's a little heart-shaped structure. And the prothallus would be gametophyte, but the bulk of the fern, the adult plant, is primarily sporophyte generation. If we look at flowering plants, the bulk of a flowering plant is going to be sporophyte. So the flowers, the leaves, etc. There are very few parts of the plants that are gametophyte. And so a difference between mosses and flowering plants or angiosperms would be that the mosses are gametophyte dominant. The leaves are gametophyte, they're haploid. If we compare that to the flowering plant is sporophyte dominant, and the leaves are going to be diploid. So that's a difference between them. So the leaves in the moss are going to be haploid. The leaves in the flowering plants are going to be diploid, right? What they have in common is they both have a sporophyte and a gametophyte generation. They both have both stages. They have a haploid part and a diploid part, but what makes them different is which part is dominant. Angiosperms or flowering plants are sporophyte dominant. Mosses are gametophyte dominant. And so we're going to look at more specific examples of how alternation of generations occurs. So for this part of the lab, I'm going to walk you through what the life cycle of these organisms look like. And then there are drawings in your lab that you're asked to draw. So for example, for the moss life cycle, it tells you to draw the sporophyte and the gametophyte on page 144. There's a little box for you to draw this. And what you're going to label is you're going to label the sporophyte and the gametophyte. So if I look at the moss, this bottom part, the green part, notice is the gametophyte. So that green fuzzy stuff that you see for a moss, that's the gametophyte generation. That is going to be this part, the gametophyte, is haploid. So when you draw this, you're going to label this the gametophyte, and that's haploid. On the flip side, the sporophyte, the diploid part, is this top portion. That is the sporophyte. And that part is diploid. So the cap and the stalk, so this is the stalk, this is the cap. And within the capsule are these spores. And within the capsule, the cells that are in there are undergoing meiosis. Because remember, this top part is diploid. So they're going to undergo meiosis, and they're going to produce those haploid spores. And then those haploid spores are going to be wind-blown. And those spores can then germinate and give rise to the early gametophyte, right? Because the gametophyte is the part that is haploid, right? It's the part that derives from the spores. So if we look at the gametophyte, we have the female gametophyte, which has something called the archegonium. And within the archegonium, we have the egg. The egg needs to be fertilized. The male gametophyte has a structure called the antheridium. And the antheridium is going to produce the sperm. Now, for fertilization to occur, we need water. The sperm is going to swim through the water, and it's going to be released from the antheridium, and it's going to get to the archegonium on the female, and it's going to fertilize the egg. And so now we have fertilization. And so within this archegonium, we're going to start to get this sporophyte embryo, because when the haploid and haploid come together in fertilization, we get back to having the sporophyte. And so from this gametophyte, you're going to get this stalk and capsule forming. And again, the life cycle is going to continue. So for the moss, you want to draw this part here, this whole part. The bottom green part, you're going to label gametophyte. It's haploid. And the top part, the stalk and the capsule, is going to be the sporophyte. So that whole part is sporophyte. And that is going to be diploid. And so this is the life cycle of the moss. If we look at ferns, remember that ferns, from the mosses, the ferns evolved to have a vascular system, which allows the ferns to grow tall. However, ferns did not evolve yet to have seeds. So ferns are a plant that reproduces using spores, but they did evolve to have a vascular system, which allowed them to grow tall. So because they only produce spores, they need to have water for sperm and egg to combine. And so when you look at the bulk of the plant for the fern, so if you look at the leaf, this whole leaf is called a frond. F-R-O-N-D. So the frond is the leaf. And the frond is going to be sporophyte, meaning it's diploid. So again, this is different than the moss where the leaves were gametophyte. In the fern, now we see that the leaves are going to be sporophyte. They're diploid. So that's the frond. On the underside of the leaves, you see these little packets, these little dots that are on the bottom side of the leaves. And these little packets of spores are called sori. And so the sori are what contain the spores, and the spores are the haploid part. Now, when the spores are released, they're going to form the gametophyte. And they're going to form a structure called the prothallus. And this structure is haploid, right? And so we have our haploid prothallus, which is the gametophyte, and then that can give rise, and I'll show you how in a minute, to the diploid sporophyte, so the big part of the plant that you typically see. So what you're going to do for this one is that you're going to draw the sporophyte on page 144. And I noticed that this page number is wrong, so ignore the 99. That was from the old lab manual. You can find where to draw that. But for this one, for the fern, you're going to draw the sporophyte. And so what you want to draw on the sporophyte is you want to draw the frond. So this is the frond. And then you want to label the sori. Sori is plural for sorus. So basically on the underside of the leaves, let's say we drew this with the underside, you're going to see these dots, and those dots you're going to label as the sori. Those are the little packets of spores. And so the way that this works in the fern is that we have its frond, its big leaf, and on the bottom side we have the sori. And the sori contain, again, the spores. And so in the sori, those cells are going to undergo meiosis. They're going to produce haploid spores. So the spores are haploid. And then the spores are going to give rise to what's called the prothallus. So this is your gametophyte. And the gametophyte, it's this little heart-shaped structure. So this is what you're going to draw. So you're going to draw the gametophyte, which we call the prothallus. And on this prothallus, we have these little nubs. And on the top, these are the archegonium, which again is going to be the female sex organ. It contains the egg. And at the bottom, we have structures that are called antheridium. And we also have rhizoids, which are used for anchoring and for absorption of water. But we also have these little nubs, these antheridium, and these are the male sex organ. The antheridium is going to release sperm, and the sperm is going to swim to the egg, and it's going to fertilize the egg. And we're going to form this diploid zygote within the archegonium. And that diploid zygote is then going to lead to this young sporophyte coming up from the old prothallus. And so that young sporophyte is going to mature into this mature sporophyte, which is going to be the frond of the fern. And so this is just illustrating the life cycle of the fern. So you need to draw the sporophyte, which is the frond with the sori, and label the frond and the sori. The frond is the whole leaf. The sori are the spores. And then you want to label and draw the gametophyte. So again, up top is the archegonium. On the bottom is the antheridium. And so you're going to draw that in your lab. Next, we have our gymnosperms. And remember that our gymnosperms are our cone-bearing plant. And remember that gymnosperms have a vascular system like the ferns. But what we start to see in terms of gymnosperms is the evolution of seeds. So these plants now are seed-bearing. They have naked seeds, meaning they're not contained within a fruit. So if we look at a gymnosperm, right, and we look at the mature tree is going to be, again, sporophyte dominant, meaning most of the plant is going to be sporophyte. And on this tree, we're going to have cones. We have female cones and male cones. The female cone is the one that we think of typically when we think of pine cones. They're the kind of hard ones. They're really big. These can be very, very large, bigger than your head. So these cones can be large. The male cones are the smaller of the two. They're very soft. They're very fragile. They're not nearly as hardy as the female cones. Now, the male cone is the cone that's going to produce the pollen. And pollen is going to be distributed by wind. And so the wind is going to carry the pollen from the male cone to the female cone. And when that pollen fertilizes the female cone, we end up with this cone that has the seeds. Now, the seed contains the diploid zygote because we have fertilization. And so the seed is going to contain the embryo. And you'll also notice that the seed has this little wing. And just like the pollen is distributed by wind, the seeds are also dispersed by wind. So it has this aerodynamic kind of tail. And so the seed is dispersed by wind. It's going to go to a new place and it's going to germinate, right? And it's going to give rise to the new tree. So for the gymnosperm, you need to draw the male and the female cone on page 143. And you need to label the female cone and the male cone. As you're going through this presentation, if you need more time to draw, just pause your video, draw it, and then push play to go to the next one. You also need to draw the seed of the gymnosperm, and you need to label the seed and the wing. So if I'm looking at this, so here's a cartoon drawing of it. Here's our seed, here's our wing. If we're looking at an actual one, this is the seed, and this is the wing. And again, the wing is there to help with seed dispersal. The seed is formed by fertilization of the pollen with the egg, and it's going to give rise to our diploid zygote, or in this case, our embryo. So the seed is going to contain our embryo encased in a protective seed coat, and that's there to protect that delicate embryo. We also have nutritional tissue that's going to provide nutrients to that developing embryo. And then the seed is going to be dispersed by wind, and the seed is going to germinate, and the plant is going to sprout out, and we're going to end up with our large diploid sporophyte tree for the gymnosperms. And so again, gymnosperms are also sporophyte dominant. So you need to draw the seed, label the seed, and the wing. Next, we're going to move on to our angiosperms. And angiosperms, remember, are our flowering and fruit-bearing plants. So we've already seen the evolution of vascular tissue. So ferns were the first to get vascular tissue. So gymnosperms and angiosperms also have vascular tissue. When we got to the gymnosperms, we saw the evolution of seeds. When we get to angiosperms, we still have seeds, but now we see the evolution of flowers and fruit. And think about what the advantage of flower and fruit are. Flowers, for example, the advantage of a flower is to attract pollinators. So different organisms are attracted to different types of flowers. If we're talking about hummingbirds, hummingbirds are a pollinator that are attracted to tube-like flowers that are yellow, for example. If we're talking about butterflies, they have a different type of flower that they're attracted to. Bees have a different one. Beetles, bats, etc. These are all examples of pollinators, and they are attracted to the plant by the flowers. So the evolution of flowers helps with pollination because remember, gymnosperms use wind to pollinate. But in the case of angiosperms, they use animals, for example, as pollinators. So flowers give that advantage. Fruit give the advantage of protection of the seed, which contains the embryo, and it gives an advantage for seed dispersal. So again, gymnosperms had to rely on wind for seed dispersal, but angiosperms can have fruit be an agent of seed dispersal because animals will come along and eat the fruit. They will also ingest the seeds. Animals are mobile. They move to a different spot. They defecate, out come the seeds, and they germinate into a new plant. So in the angiosperms are going to have the evolution of flowers and fruit. So what you're going to draw for the angiosperm, and this is on page 144, it says draw the flower. You need to draw this flower, and you need to label the following parts. So the stamen, the way I remember this, the stamen is the male part of the plant. And that is that the anther contains the pollen. And the pollen are the male gametes. So this little yellow structure here at the top is referred to as the anther, and that contains the pollen, which again are the male reproductive cells. Also part of the stamen, the part that holds the anther out is referred to as the filament. So the filament is going to hold the anther up. Now, the reason for that is that the pollinators need to brush up against the anther to pick up the pollen. And then they're going to take that pollen to a new plant. So the filament kind of holds the anther up, and it allows the pollinator to come in contact with that pollen to be able to transfer the pollen from one plant to another. Now, also on the flower is the female part. And the female part is called the pistil, or it's also called the carpel. They're kind of used interchangeably. And so if we look at the female part, we have what's called the stigma. And the stigma is sticky, allows pollen to stick. Because the pollen has to land. The pollen has to land on the stigma. And so the pollen's going to stick, and then the pollen is going to travel down through this pollen tube, this tube, within what's called the style. So the style is going to hold the stigma up. It's this narrow part here. And so the pollen is going to travel its way down the pollen tube, and it's going to come to the ovary, right? And the ovary, think of female reproductive organ in humans, is also referred to as the ovary. And within the ovary, we have the ovule, which is the egg. So the pollen is haploid. The egg is haploid. The pollen is going to fertilize the egg, and then that fertilization is eventually going to result in the seed. So you need to label the female parts of the flower, which are the pistil or the carpel. So label the stigma, the style, and the ovary. You need to label the male part, which is referred to as the stamen. So make sure you label it stamen and male. You need to label the anther and the filament. Some flowers are what are called complete flowers. They have both male and female reproductive parts on the same flower. And other flowering plants are incomplete, meaning you have a separate plant for the male and the female. It doesn't have both in the same plant. But this example that you're seeing here is a complete flower. It has both male and female in the same plant. The petals, so you want to label the petals. We have this receptacle, which is going to hold the ovary in place, etc. So you need to draw this and label the parts. And so this is just showing you the life cycle of the angiosperm. So again, in an angiosperm, the sporophyte is the dominant part of the life cycle. So the leaves are sporophyte, the flowers are sporophyte, etc. So within the mature sporophyte, we have the female part. So again, the pistil, and the male part, which is the stamen. Men is male. And so the stamen has the anther, and the anther contains the pollen. And the pollen, again, are the male reproductive cells. And so this cell, these cells within the anther, are going to undergo meiosis. They're going to form the haploid pollen. And in the female, in the pistil, the cell within the ovary is going to also undergo meiosis and form the haploid egg. And so the sperm cell in the pollen is going to land on the stigma. It's going to travel down through the pollen tube, and it's going to fertilize the egg, and it's going to form this diploid zygote. Now, interestingly, in flowering plants, they do something called double fertilization, and that is that there are actually two sperm within the pollen. One is going to fertilize the egg to give rise to the embryo or the zygote, and the other one is going to form the nutritive tissue. And so that's just called double fertilization. I won't ask you that, but just be aware that that happens in angiosperms. And so now during fertilization, we go back to having our diploid sporophyte. It's going to form the embryo. The embryo is going to form within the seed. And then the seed is going to be eaten, let's say, by an animal, which is going to walk and it's going to disperse the seed. The seed is going to germinate and it's going to give rise to the mature sporophyte. And the process can keep continuing. And so this is kind of how the life cycle of the angiosperm works. Now, the fruit is the mature ovary of a flowering plant. And the fruit is going to be the fleshy, spongy matter that protects the seed. The fruit not only functions to protect the seed, but also, again, aids in dissemination of the seeds so that they can germinate. Again, because it attracts animals. Animals are mobile. Animals eat the seed, and then they move and defecate them. And now we get a new generation. If we talk about the fruit, the pericarp, that is the part of a fruit that encloses the seeds and develops from the wall of the ovary. And the pericarp can be divided into three parts. The exocarp, which is the exit, think of outside, it's the outer layer. The mesocarp is the middle. So if we're looking at an orange, for example, that tough leathery rind is the exocarp. The mesocarp is that kind of spongy material in the middle. And then the endocarp is the part that we typically eat. That's the fruit that we eat. And so that's the endocarp. And so those are containing the seeds. So there are several types of fruits that can happen. We can have simple fleshy fruit, what we call a drupe. So an apricot, a peach, a plum, etc., where we have one carpel and one seed. So this would be an example of a simple fleshy fruit. So an apricot, for example, has one carpel and one seed. We can have what's called a simple dry fruit. So a pea pod would be an example of this. It has one carpel. So notice that that's the wall of the ovary. And within it, we have many seeds. And they're linked to this pod by a placenta, kind of like we have a placenta. And we can have what's referred to as a compound fruit. In the case of a strawberry is actually a false fruit. And that's because the seeds, those little seeds on the outside of the strawberry, each of those is derived from a carpel. So when you look at the strawberry, it has many seeds, it has many carpels, but in this case, the fleshy part that you eat is actually not from the ovary like it is for the others. The fleshy part of a strawberry is actually from the receptacle part. And so that's why we call strawberry a false fruit. It's not actually technically a fruit in the sense that the fleshy part is not derived from the ovary. The fleshy part in this case is derived from the receptacle. So what you need to do for this part is to draw the fruit on page 144 and label the seed and the ovary. And so for this, I gave you several types of examples. So one example, you can see it here. This is an apple. And so the apple, where the fruit is going to happen. It's going to form around the seed. So if we look at this other labeled diagram that I have put here for you. So if we look at the apple, this is the mature ovule, which is the seed. That's the seed. And the ovary is that kind of outline that you see within the apple. And so that's the ovary. So you're going to label the ovary, this line, and you're going to label the seed. If you're looking at the tomato, the seeds are inside. This is actually a fruit. So a good rule of thumb is if it has seeds, it's classified biologically as a fruit. So it has its seeds, and then the ovary is going to be this whole part here, this pericarp, the endocarp, the mesocarp, and the exocarp, which is the outer part of the tomato. And so that would be where you would label the ovary. If you're looking at a peach, for example, right? So in the middle, the hard pit is going to have the seed, and the outer part of that pit is going to be the wall of the ovary, which again is going to be our pericarp, our endocarp, mesocarp, and exocarp all combined. And so you need to label and draw one fruit and then just label the seed and the ovary. And so the last part of this is not looking at plants anymore, but instead looking at fungi. And remember that our fungi are our molds, mushrooms, and yeast. And we're going to talk about how fungi reproduce. So the first thing that you're going to draw is rhizopus. And rhizopus is black bread mold. And so rhizopus can reproduce either sexual or asexual. If they reproduce asexually, they form a structure called the sporangium. And so what you're seeing is you see these little filaments that are coming up, and then you see these little black balls. Those little black balls are structures that are called sporangium. And within the sporangium are the spores that are used for reproduction. Notice that from the sporangium, you have one little line coming up to it. That little line is referred to as a hyphae. So the hyphae is the diploid part, and then it's going to give rise to the spores. Now, the spores are within the sporangium. The reason you know this is asexual, notice there's only one hyphae going to the sporangium. There's no partner. That's what tells you that this type of reproduction is asexual. On the flip side, rhizopus can also form a structure that are called zygospores. And so you'll notice here's my spore, and I have two hyphae coming off of each of my spores. Here's a hyphae, here's a hyphae. Hyphae, 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 hyphae. So zygospores are sexual reproduction for rhizopus, meaning that these two hyphaes come together to reproduce in this zygospore. And within the spore are going to be the reproductive cells, but that is going to be sexual reproduction. So you need to draw the sporangium, which is the asexual, label it asexual, and you need to label the zygospores and label that sexual. And again, look for the number of hyphae going to those structures. If there's one hyphae, it's asexual. If there's two, it's sexual. And then the last fungi that you're going to draw is going to be a mushroom. And so you're going to draw a mushroom on page 145. And there are several parts that you need to label. You need to label the cap. So that's this top part here. The cap. You need to label the stalk. That's the stalk. The gills. The gills are those little lines. You can also see them there. The gills are the little lines up in the cap. Now, within the gills, you're going to find spores. And so there's these little spores in between the gills. So you can just draw, if you're drawing this drawing, you can just draw and label that there are spores within the gills. And so this is looking at a cross-section under the microscope of the gills and the spores. So you can see the lining of the gills, those little openings there. And you can see those little dots within there. Those are the spores. And those spores are used for reproduction. And so this bottom right picture is what you want to draw for your mushroom. Label the cap, the gills, right, these little lines, the spores, the dots within the lines, and then the stalk. That's your fungi. The last part of this experiment is looking at the effectiveness of hand washing. And so bacteria, right? Bacteria are prokaryotic cells. They're very simple. They have, in many cases, one circular chromosome. Remember that they don't have membrane-bound organelles. They do have ribosomes because ribosomes are used for protein synthesis. They have cell membranes and they have a cell wall. So in this experiment, we would be testing the effectiveness of hand washing. And so the way this would be done would be some people would choose to wash their hands. Some people would use different types of hand sanitizers to look at how effective hand washing really is. Now, this again is very relevant to what's going on in the world right now because we are talking about the effectiveness of hand washing for viruses. You need to effectively wash your hands to prevent transmission of the coronavirus. Now, when doing this type of experiment, doing this with bacteria is much easier to work with than trying to test for viruses on your hands. The reason is that bacteria are free living organisms. They reproduce on their own. They don't need a host cell. So I can take bacteria and put it on a nutrient agar plate and it will grow. If I was looking for viruses on my hand, that same approach is not going to work because viruses are obligate intracellular parasites. They have to reproduce within a host cell. And so it's much more difficult to look at the viruses on your hand than it would be to look for the bacteria. But looking for the presence or absence of bacteria will also give you an idea of how effective your hand washing is. Because if your hand washing is effective, you would expect to see a reduction in the number of bacteria. And so on each square centimeter of your skin, there are about 1,500 bacteria. And so if you look at your hands, even within your two hands, you have very different, what we call normal flora or normal microbiota, meaning different types of bacteria on your left and your right hand. So let's talk more about this. So there are several ways that bacteria transfer can happen onto your hand. One way is after handling raw chicken. Raw chicken is often going to have a variety of different bacteria, ranging from salmonella, for example. And so if you handle raw chicken, you want to be conscientious about not touching other surfaces in your kitchen. Because if you do that, if you touch that raw chicken and then you continue to touch things all over your kitchen, let's say you open the oven with that hand, well, that bacteria on your hand is now on the oven. And so it's important that you wash your hands after handling raw chicken, basically to prevent transmission of the bacteria from your hands onto other surfaces. After handling an old dishcloth, because oftentimes those dishcloths are wet, they're moist. If you didn't wash your hands well and you touch that dishcloth, well, bacteria is now on that dishcloth. And so if you keep using that dishcloth over and over and over again, when your hands weren't all the way clean to begin with, well, you're going to end up with a bunch of bacteria all over your hand. After using the toilet. So if you go to the bathroom, you probably know that you should always wash your hands after going to the bathroom, because even if you think you wiped and you didn't transfer any bacteria, there's no way to know. So you should always wash your hands after using the toilet. The other thing to think about in terms of using the toilet, this is something that I always like to get students to think about, is let's say you have a bathroom, and in your bathroom, your toilet is right next to your sink. And you store your toothbrush in one of those holders on the counter, on the sink, on the counter of the sink. If you have one of the bathrooms where your toilet is right next to your sink counter, your bathroom counter, and you flush the toilet, what is effectively happening is that the bacteria is being aerosolized. The bacteria is actually going up into the air and onto your toothbrush. And so it's a good idea if your toilet happens to be in the same room, especially as where your counters are, close the lid. Close the lid of the toilet and then flush. That will help prevent aerosolization, preventing those bacteria that's in feces to become airborne and end up on your toothbrush. That's just gross to think about, right? So you want to think about those things. After cleaning a surface used for meat preparation. So again, let's say I had cooked chicken and I now clean my cutting board and my knife and all those things. Well, after I'm done cleaning those surfaces, I also want to wash my hands because I'm still going to have bacteria on them from cleaning those surfaces. And so it's a good habit to get into proper hand washing, proper transfer, touching things, and then washing your hands. So the way that this experiment would have been done is you would get 2 plates. One person would do this experiment. So you would do this in pairs. One person would do this experiment. And so what you would have is you would have a bottle of sterile water, and you would take your sterile swab and dip it in the water. And then you're going to take that swab and you're going to swab your hand. Now, a couple things to keep in mind when you're doing this. Like any experiment, you need to have only one variable, meaning only one thing that changes from one plate to the next. And so what I mean by this is if I do my before hand washing plate with my left hand, I need to do my after my hand washing with my left hand. Because again, my left and my right hand are not necessarily going to have the same bacteria. So you always want to think about when you do an experiment, consistency. Only one variable. And the one variable in this case is going to be before hand washing and after. So you have to use the same hand. You want to swab all the same parts of the hand. So if you swab between your fingers, for example, on your before hand washing, you better be able to also swab between your fingers on the after. So I dip my swab in the water and I swab my hand. I run the swab all up and down my plate. Or I'm sorry, my swab all up and down my hand. Now, when I swab my plate, let's say this is my before. Just to give you an idea of how this works is these agar plates are very soft. Think of them like Jell-O. If you're going to swab, you're going to take your swab and put it on your agar plate. You would take your swab and gently start at the top, back and forth, back and forth, back and forth, all the way down the plate. You want to make sure to cover the entire plate. So I tell students, go back and forth, back and forth. Go side to side. You'll notice that if you're going side to side, it's not easy to get to the bottom of the plate. Because in order to do that, you would have to put your swab straight up and down. And that would put too much pressure on the agar and it would tear. So what I tell students to do is back and forth until you get to the bottom where you can no longer swab without putting your swab straight up and down. Then what you would do is you would rotate your plate 90 degrees so that now when you swab, so let's go back and put red back. So if I rotate my plate 90 degrees, my plate is going to look like this because I've moved it over 90 degrees. So that now when I swab, so here's my purple, I go side to side, side to side, and that gets this other portion that I couldn't get again. And then I would rotate my plate 90 more degrees, and that would allow me to get this top portion. So when you do this, it's really important that you swab all parts of your plate. So I swab from the top, I work my way down. When I get to the bottom and I can't swab side to side anymore, rotate my plate 90 degrees. So now that top portion is off to the side, and I go again from top to bottom, side to side, side to side. When I can't get any further down to the bottom, rotate my plate 90 degrees. So again, this bottom part is over here. And then again, I swab side to side, side to side. So that would be your before hand washing plate. Then you would use whatever method you were testing. So some of you might test like the soap in the classroom. Some of you might test dial soap. Some of you might test Purell hand sanitizer. Some might test Target hand sanitizer. And so whatever method you're testing, you would use that. So you would use, let's say, the Purell. And so you would put the Purell on your hands, right? And give it time to kill off any bacteria. And then you would repeat the experiment. So you would get a new sterile swab. You would dip it in the sterile water. Then you would take the same hand that you used before. So again, if I use my left hand for the first plate, I need to use my left hand for the second plate. And I'm going to swab my hand after my hand sanitizer. Again, in the same way that I swabbed it the first time. So if I went between my fingers, in my after, I need to go between my fingers again. So I'm going to swab my hand. And then I'm going to repeat the same process on a plate that I label after. So I have my before hand washing plate and I have my after. And so we would take those plates that have agar. Agar just is a nutrient-rich food source for bacteria. And I would take those plates, my agar plates, and we incubate them agar side up. And that's because bacteria will produce condensation. And we want the condensation to be on the lid and not on the agar. So we incubate them agar side up and we put them in the incubator at 37 degrees. Think about why we test this at 37 degrees Celsius. What does that temperature correspond to? And if you said human body temperature, you are correct. That corresponds to human body temperature, which is what we want to see the growth of because we're testing it on our hands and our bacteria are on our hands are at human body temperature. So next week, we'll talk about the results of this experiment, what the plates would look like before and after hand washing. And so this is what we would do. So the first video that I'm going to show you here is a video that shows you how to properly wash your hands. And so in this video, they're using the purple paint as soap to show you how to cover all the surfaces of your hand with the soap. So this is a good lesson for how to properly wash your hands.
Let's first put soap in the palm of our hands and then begin to rub in between like so. But as you see, there is no soap on the back of my hands, so I'm going to interlace my fingers palm to palm, and then I'm going to do the same technique on the back of my hands with each hand. And then you're going to take the back of your fingers to opposing palms with fingers interlocked. And then start rubbing your thumbs. And then you're going to take your fingertips, clasp the fingertips in the right hand in the left palm, and vice versa. Rub your wrist one last time to make sure your hands have been completely covered in soap. Thanks for watching.
This video is looking at cross-contamination. And so this is, again, especially relevant to what's going on now about COVID-19 and people wearing gloves when they're out. And if you're not using them properly, you're actually not doing yourself any favors. And so this is going to illustrate this to you to show you what are all the problems with not using your gloves properly if you're using them.
Hi friends, Professor Ashley here, coming with a visual demonstration to once and for all stop the madness. To answer the question, to glove or not to glove? That is the question. To glove or not to glove? Now we've all seen the controversy lately on social media about whether or not we should be wearing gloves in this time of COVID-19. And I'm going to answer that question for you because it's a multifaceted question. So we've all seen so-and-so putting on their gloves and then walking through the grocery store, aisle to aisle, item to item, to the cash register, then out and into their car wearing the same pair of gloves. Now, here's what's important. We're not going to judge, okay? Because as I like to remind my students, we are only as good as what we are taught. We are only as educated as our education. So, as an educator of nursing, I'm a huge fan, and my students can attest to this, of visual demonstrations. So I'm going to give y'all a visual demonstration to answer the question of whether or not you should glove or you should not glove. So, here's our visual demonstration, a hypothetical situation. I'm going to put on my gloves. And I'm going to go into the grocery store with my gloves on, right? I'm going to try not to touch my face, all these things. Got my gloves on, well protected. I'm only going in for 2 or 3 items. No big deal, shouldn't be long, okay? So I'm going to go, first of all, I'm going to touch a cart. Now I touch the cart. Yep, means it's on this hand too, touching the cart, rolling through the aisles of the grocery store, okay? We found toilet paper. We're lucky today, people. Let's pick up the toilet paper. Have a look at the toilet paper. Yep, looks good to me. I'm going to put that in my cart. I'm going to put that toilet paper in my cart. Oh, excuse me just a second. Somebody's sending me a text message. Let me just check my phone here. Oh, it's just my partner. We need chicken. No big deal. Yeah, I got the chicken. Don't worry about it. I'm going to take my phone now. I'm going to put my phone where? In my purse. Phone's in my purse. Let's carry on. Oh, man. And if you wear glasses, you know, glasses just drive you crazy, right? My hair's not really staying where it's supposed to be staying today. Got the toilet paper. We're good to go. Now let's mosey on down. We need chicken. Just called, said we need chicken. Oh, there's so many different kinds of chicken to choose from. I don't like cutting chicken, so I'm going to just get this chicken breast. We ain't got to cut that. Got us some chicken. Okay, good. Oh, 1 second. My mom's calling. Hello? Yep. I'm at the grocery store. Do you need anything? You got TP? Yeah. Okay. Yeah, I can pick that up. You need anything else? Hang on a second. You need something else? No? Just that? Those 2 things? Okay. Yeah, no problem. I'll put it on the porch. I got you. Good. Good stuff. Okay. Talk to you later. Love you too. Bye-bye. I'm going to just put it in my pocket since everyone seems to need to get hold of me right now. I'm going to just put that in my pocket. Shampoo. We need shampoo. Let's go look at the types of shampoo. Pantene, I don't know. L'Oreal. There's so many different types of shampoo. All these types. How's this one smell? Oh, that one smells like coconut. I like that shampoo. We're going to get that one. We're going to get the coconut smell shampoo. That sounds like a good idea. Good. Oh, my phone again. Excuse me. Jeez. Just another text. I'm at the grocery store. Leave me alone. Oh, my kid's getting out of hand. I should probably let them play with my phone just to keep them in line while I finish up my grocery shopping. Here you go, honey. You play on Mama's phone while Mama finishes grocery shopping. Okay, no problem. Let's continue on. I think that's it. I only needed like 4 items, right? We're good. We're good to go. We're good. We got this. Okay. This, everybody, this is a visual representation of cross-contamination. This is what cross-contamination is. And this is literally as simply as the majority of bugs are spread from surface to surface and therefore from person to person. Okay? So, to glove or not to glove? Are gloves bad? No. Are gloves a replacement for washing your hands? Hard no. Also no. Let's not forget that by the time we then go to the checkout, put our hands inside of our purse, get our money, pay the person, you might as well do this. And you might as well do this. Yeah, that just happened. Okay? While I'm on the topic of cross-contamination, there is one way and one way only, which is the proper way to take gloves off and dispose of them. Okay? If you put this glove anywhere on your skin, contaminating, which obviously I've done by touching my face as a visual representation, okay? One correct way. You take your non-dominant hand, pull your fingers out, okay? Grab that glove, put it in here. Now take your clean hand, put it inside of the cuff so you're never touching the outside of the glove, and pull it off, okay? Dispose. This is the inside of the glove. I'm never touching the outside of the glove. There's only one correct way to do that. Following taking off gloves, if you manage to use them without cross-contaminating, you absolutely need to wash or sanitize your hands. Okay? So what is the take home here? Wash your hands. This is why you keep seeing and hearing, wash your hands, wash your hands, wash your hands, wash your hands, wash your hands. Okay? Wash instead to stop the spread. If you're going into the grocery store with a pair of gloves on and you're touching more than one item, which you are, and you're not changing your gloves in between or washing those gloves or sanitizing those gloves, you aren't doing yourself or any of your loved ones any favors. Okay? So I hope this messy visual representation of to glove or not to glove will help y'all. And with that, I'm going to go wash my hands and my face. Thank you so much for watching. Tune in for more at Empowerment NCLAC/CPNRE CPNRE Test Prep at any time, or join us on our YouTube channel. Stay safe and stay sanitized.
And so that just shows you the power of cross-contamination. And so hopefully that illustrated things not to do when you're at the grocery store if you're trying to protect yourself from the coronavirus. So I just wanted to address a little bit about these questions. So question number 2, describe the similarities and the differences in the sporophyte generation and the gametophyte generation between mosses and flowering plants. And so again, you want to go back to the slide where I was discussing alternation of generations, because I did go through and I talked about what is similar between mosses and flowering plants and also what is different. So refer back to that slide in that part of the video when you're trying to answer question number 2. Question number 3, what did you expect to be the actual number of people who were infected by an STI after 2 contacts? What about 4 contacts? So not just what do you guess off the top of your head, but what was the expected number using that formula that we talked about? So what was it after 2 contacts? What did we expect after 4? Question 4, was your actual number of people who were infected after 2 or 4 contacts different than what was expected? If yes, why do you think some people were not infected? And so this is what I went over with you, right? So when we looked at our 4 contacts, for example, was it the same as we expected? If not, what were some of the reasons that I talked about for why people would not be infected? Why our actual would be lower than our expected? So make sure to address both parts for that question. And then the last question is to research 2 sexually transmitted infections. Make sure that one is caused by bacteria and one is caused by a virus. So again, make sure one is a bacteria, one is a virus. And so the first column for this says STI name, so the name of the disease, and then the type is referring to if it's the bacterial one or the viral one. You want to indicate how you get the STI, the sexually transmitted infection. So notice it has a column for method of infection. Do not just tell me sex. Be specific. What types of sexual contact can transmit the disease? Because it might not always be the same. What are the symptoms? Again, don't just say that there are no symptoms. They might have some people be asymptomatic, but other people will have symptoms. And what might those symptoms look like? What are the treatments, and if there's a cure or vaccine for each sexually transmitted infection? And so again, if you say treatment, don't just say antibiotic, for example. Give an example of an antibiotic. So what specific antibiotic? Like the doctor could prescribe penicillin or azithromycin. What is the actual antibiotic that they might prescribe? Is there a cure? Is there a vaccine? Sources used for your research. So the Center for Disease Control, the CDC, is a good source to use. Make sure you're not using sources like Wikipedia. That is not a reliable resource. Mayo Clinic might be a good one to use, CDC, possibly the World Health Organization, etc. Make sure it's a reputable source and cite where you got your information from. And so again, one should be bacterial, one should be viral, and you need to fill out both rows, one for the bacterial and one for the viral. And so again, be specific. Don't just say sex, don't just say antibiotic, be specific. So the last thing is just to talk about the Quiz 10 study guide. So advantages and disadvantages of sexual and asexual reproduction. Understand alternation of generations in plants, which phase is haploid and which phase is diploid. Be able to identify pictures of various organisms that you drew. So to be able to identify if you're looking at a moss or a fern, are you looking at the gametophyte generation or the sporophyte generation, etc. Be able to identify the difference between the male cone and the female cone. So if given a picture, you should be able to tell me if it's male or female. Identify the parts and the function of the flower parts. So you should be able to label or know where the anther is and that the anther contains the pollen. And the stigma is on the pistil, and it's the sticky part where the pollen lands, etc. So identify the parts and the function of the flower parts. Be able to calculate the expected number of infected individuals if given a number of contacts. So if you were given that somebody had 4 contacts, the expected number of infected individuals should be 2 to the 4th power. So 16 people would be expected. Know reasons why the expected number of infected individuals does not always match the actual number. And so just make sure that you go through and you review your slides as well as your lab write-up to help you study for this. Make sure when you study your slides that you're not only studying the plants, but also to be able to identify the fungi as well. So like for the rhizopus, know the difference between the sporangium and the zygospore. Be able to identify which is asexual, which is sexual, etc. And then read through lab 11, looking at development. And so this is the end of lab 10. The one last thing that I want to say is for this lab, you are not required to do the graph. For this one, no graph required. So that's the one exception to this. No graph. You do not need to do the graph for this.